Okay, now we're talking about Chapter 2. 2 is delving in just to the innate immunity system. So the chapter overview for the innate system, Chapter 2, is external defense systems, which are the physical, chemical, biological barriers that we have, and then the internal ones, in other words, deeper. PRRs, which are called pattern recognition receptors, the acute phase reactants, things that tell us that something's going on, the whole inflammatory process, what is phagocytosis and how we kill our pathogens, a little more in depth on the natural killer cells, and the different innate lymphoid cells. So the innate immune system, it acts immediately once a pathogen gets into our body. It's very nonspecific. Just about everybody has it. It works extremely quick. It's in minutes, seconds for it to happen. So there's external and internal different types of defense mechanisms that we have. Most of the external that we have are basically what our body has set up for us to prevent things from entering our body. So it's more of an anatomical type stuff, and we'll see that here in a second. The internal is dealing more with a cellular response and how we can recognize certain components of the pathogen itself. So the external defense system, you can see from the picture there are physical, chemical, and biological barriers that are out there that prevent, any kind of infectious agent from getting into the body. These antigens we're talking about. For instance, the salivary and lacrimal glands that we have, they have digestive enzymes in there to help break down any kind of foreign thing that gets there. Our nose, we cough and sneeze. You've had stuff go the wrong way. You know how that happens. But anytime you're exposed to things, coughing and sneezing can expel some of the microbes that we have there. Of course, in the airway, we have mucus and cilia that keeps stuff. Cilia, expelling mucus, how you cough and cough things up. Mucus can trap the microbes so they don't get anywhere. The skin itself has got cells called keratinocytes that actually will keep bacteria out. That's why it's important that the skin is maintained so it's a good physical barrier. The sweat glands themselves have lactic acid built in them. And then the sebaceous glands, which, they call it the sebum, like the oil of the skin, I guess you could say. It has like fatty acids in it. Then the stomach itself has a gastric acid in there. It's like a pH of 2. It does a really good job. It's amazing stuff can still penetrate through it, but it does a good job of taking care of most things. So the skin, the epidermis itself is epithelial cells, if you remember that from A&P. They're actually coated in that keratin that we were talking about. So it keeps the pathogens from coming in. The dermis itself is a connective tissue that has blood vessels in it, that has, hair, the sebaceous glands in it, sweat. That's where the white cells will be, the monocytes, and sometimes the neutrophils will be located there to protect. And then, like the secretions, we talk about sweat, tear, saliva, all that kind of stuff, to try to keep the pH more of an acidic thing there. And then there's proteins in there that are all antibacterial to protect it. So that's why it's such a big deal to keep the skin intact, to make sure we don't get any kind of infectious process to go through it. And if it is broken, that we clean it quickly, antibiotic ointments, whatever, Band-Aids, that kind of stuff to make sure. The mucous membranes, you don't want those to dry out. They got to stay nice and moist. They line the respiratory, urogenital area, and digestive system. And again, they are another physical barrier that's there. The mucus secretions keep stuff from not able to penetrate in there, and it will block it to make sure. So in other words, it'll shed it off, so it won't allow it to adhere to the epithelial cells themselves. Respiratory tract actually has little finger-like projection cilia that's in there that keep stuff, from coming in deeper. They're sort of leaning in one way, so that the bacteria can't, or virus can't get in deeper. That's when you start coughing and sneezing. to try to remove any kind of pathogenic stuff that gets in there. So we're talking about pathogens, meaning bacteria, virus, fungi, anything that can cause a disease in our body. The digestive system, the hydrochloric acid with the pH, it's normal like 2 or so, but it can't go as low as 1. So it will prohibit a lot of microorganism growth itself. Now, some microorganisms have toxins that can go through that would cause like food poisoning, like Salmonella, Staph aureus can do it, Shigella, those different ones out there. So tears, you've had some get in your eye, how it floods it out quickly. Saliva, will help us attack cell walls. That's why a dry mouth is a big deal. Dry eyes is a big deal. You need that external defense mechanism to help protect your body. The GU tract, just urinating, can keep bacteria from growing because it's more acidic, so it will force it out. I'm sure you've all heard about drinking cranberry juice to make sure to keep the pH acidic. Of course, you would need a lot of cranberry juice to do that kind of thing, but you need that. The vaginal canal, maintaining an acid pH also. So we have on our skin in all these areas, normal flora, microbiota, it's called biota, either way. And that helps keep pathogens from being there. You've heard, people all drink or eat probiotics, can't think of the word here, yogurt, that kind of stuff, to make sure we keep that bacteria that we want in there, because that keeps the bad stuff from growing in there. So it does a great job. The internal stuff, that's when we're talking more about cells and all the liquid stuff, the soluble factors that come out that. Remember, they're still non-specific, and that will protect us. So they are called PRRs, which are called pattern recognition receptors out there. Acute phase reactants, the inflammatory process, the phagocytic cells, natural killer cells, and the innate lymphoid cells out there. And so we'll talk about each one a little bit. So PRRs, pattern recognition receptors, they are found on the macrophages, the dendritic, the neutrophils, the eosinophils, monocytes, mast cells, T cells, epithelial cells, all those cells, which are called antigen-presenting cells. So what they do is they recognize molecules or things on the infectious stuff coming in to tell us it is an infectious thing. It doesn't belong on us, And these things that are on the microbes themselves that tell us that they're foreign things are called pathogen-associated molecular patterns. They're called PAMPs. So the PRRs recognize the PAMPs, okay, the PAMPs. You can say it either way you want. I don't care. So when the PRRs are bound to the PAMPs, this will activate these phagocytic cells, which we just said the PRRs are on the macrophages and the neutrophils and the eosinophils and the monocytes, blah, blah, blah. So some examples of, these PRRs are TLRs, CLRs, RLRs, and NLRs. Too many LRs here. So the TLRs are the most common ones that we deal with. They're called toll-like receptors. So it's recognizing certain things. And you can see there's one for gram-negative, there's one for motile bacteria, there's certain ones for mycoplasma, certain ones for funguses, certain ones for mycobacteria. So they're toll. In other words, they're looking for certain things on the cell surface or in the cytoplasm. So what happens is these glycoproteins will bind to the substances on the microbes themselves, and then it'll activate chemicals so that they can then cause more cells to come or activate other immune responses that we need. So it can cause cytokine or chemokine responses. And what that does is tells the body, hey, these need to be eating, so that will enhance phagocytosis, and that will then trigger the more specific, the adaptive system to start working, because we want to destroy this pathogenic, this stuff, these antigen stuff, before actually a disease could possibly set in. And you're probably thinking, like, if all this is going on, how in the world do we get sick? Well, something falls through, something breaks through, the immune system gets suppressed, and we'll talk about that a little bit later too. So other PLRs that are out there are called C-type lectin receptors, which are the CLRs. There's that ones that actually bind to mannan and beta-glucans, and they're particular to the fungal cell wall. So they'll activate cytokines, chemical talkative reagents out there, soluble substances out there. The RLRs, retonic acid inducible gene I-like receptors, now you know why we call them RLRs, they actually recognize RNA from RNA viruses. So they're specific for that. So that's how I sort of remember it. RLRs are for RNA. And they will induce the cytokines and a type 1 interferon. And interferons is another type of cytokine. In other words, it's a chemical that our body produces so it can talk to other cells and say, hey, you need to do something. And then the NLRs are nucleotide binding oligomerization domain-like receptors. NOD receptors they call. So they're after certain particular nucleotides that are seen in certain foreign antigenic kind of stuff. Alright, and you're probably thinking to yourself, good grief, I have never seen so much stuff in my life. But it's very specific, We're getting more in depth into the cellular responses of stuff. So the liquids that, we see, not the cytokines, but other liquid stuff that comes out, we call them acute phase reactants. That's why they're called soluble factors. In other words, they're in the liquid stuff, which means they're found in the serum, which is a liquid portion of our blood. And when somebody has an infection or something is hurt or injured or traumatized, we'll get more soluble factors out there. And what this does, it helps our body to initiate contact between the microbes and phagocytic cells. So it sort of tells the phagocytic cells, with these factors out in a generalized area, like, hey, wake up, start doing something. We need you to start destroying stuff. Now this will limit the host destruction process because the proteolytic enzymes are released because of the phagocytosis. So we just sort of focus it on certain areas. So it will bind and recycle important proteins that we use in inflammation so that we reuse them because they're important. I mean, this is, one of the tiny steps that is needed in the inflammatory process to protect our body against any kind of foreign thing that gets in that needs to be destroyed. So some examples of the acute phase reactants that are out there. C-reactive protein, CRPs, which we'll be doing in lab. Serum amyloid A, alpha-1 antitrypsin, fibrinogen, haptoglobin, ceruloplasmin, and complement C3. Now, you really don't need to know the response time per se, but you can see they're all pretty quick. CRP is the fastest, of course. And the concentration, that's not important. And you can see that they increase pretty hefty amount. So some of them you sort of remember, or you're like, I recognize from hematology, like fibrinogen and haptoglobin, that kind of stuff. But CRP is important in activating complement. Now, complement is a whole series of proteins that are out there that will actually latch onto like a cell, and they'll latch onto this foreign thing in particular that we're after to destroy, and it actually goes through all these different steps, which we'll talk about in another chapter, and actually will blow a hole, like it drill a hole into a cell wall and destroy the cell, this foreign thing that we want to get rid of. So CRP's whole function is called activating the complement, which what we just mentioned, or is called opsonization. Now, opsonization is just what our body uses to say, hey, this needs to be destroyed. For instance, an example I like to use a lot of time is, say you're on the highway, you're on 95, and you're starving, and you see those golden arches up there. You're like, oh, I know where to go now. So that's basically what opsonization is that we have labeled this foreign thing that this is what needs to be destroyed. So instead of like milling around looking, CRP allows us, targets stuff, say, hey, this is what needs to be destroyed. So it speeds that process up. And you can see the rest of them, activates monocytes and macrophages, like binds to hemoglobin, clot formation, binds to copper. So you can see all of them are a big process, part of that whole process for inflammation. So their acute phase, in other words, just when inflammation starts, that's when we'll start seeing increases in this stuff. So the whole process of inflammation, and you've all had it happen to you and you see it, is the body's overall reaction to when we get injured or invaded. Alright? And you'll cut yourself. I mean, doesn't it get red? Well, it's getting red because there's an increase of blood flow to the area. That's called erythema. And it swells because there's so much blood there. You know, it's swelling up and it causes edema. And it's hot because there is a lot of blood there. So, you know, we got 98.6 Fahrenheit or 37 degrees centigrade causing it. And it's swollen so it hurts. You know. So that's the cardinal signs of the inflammatory process that we have. So here's just looking at some major events that are involved in this inflammatory process. So if you look at step 1, this is where the macrophages or mast cells, but let's just stick with macrophages right now. Macrophages will release, you know, at the site of infection, a chemokine, a chemical communicator that will cause the blood to dilate, vasodilate, and induce selectins. What will happen is these selectins will bind to leukocytes and tell them to start moving on the vascular wall. Like, you know, wake up, we got something going on, I need you to go out to the tissue and eat this crap. Basically, you know, that's what we're talking about. So now, you know, because the chemokine-induced integrins are on the leukocytes, that will allow the leukocytes to like squeeze through the endothelial tissue, the endothelial cells. You know, and that process is them squeezing through is called diapedesis. It's like they're moving at a slow process to go right through the tissue to go to the site of infection. So once the leukocytes are out into the tissue, they actually will follow these chemicals, these chemokines, that remember the monocytes, the macrophages, the dendritic cells, and all that stuff is released. And because they're moving because of these chemicals being squirted out, they call it chemotaxis. So it's a chemical taxi, and it sort of directs them to the area where the infection is so they can start doing their stuff. So once they get there, they got to go through the process of phagocytosis. So the phagocytic cells go to the area of inflammation. So remember, we talked about the phagocytic cells being the neutrophils, the segs, the monocytes, the macrophages, the dendritic cells. So this process will be really enhanced if the C-reactive protein, the CRP, is actually stuck on there. That's the opsoning. In other words, targeting it, labeling it to be destroyed. So in that first phase of phagocytosis, you can see that there is adherence. That's where there is actually contact between the phagocytic cell and the microorganism. Now, if it's been labeled, targeted, opsonized, that speeds the process up because it knows where to go real quickly. Alright, so the next phase is, you know, we have adherence, so it's attached, physically attached. Now the second phase, as you can see the picture, is engulfment. In other words, the cytoplasm of the phagocytic cell now surrounds the microorganism. So then you go to step 3, so that's where we actually form a phagosome. So now the microorganism is actually in the phagocytic cell, and it is surrounded by a part of the cell membrane. They call that a phagosome. Now, once we have that phagosome inside of the cell, of the cytoplasm of the phagocytic cell, there will now be little granules that we had that remember we saw in the neutrophil, eosinophil, so forth, so on. These granules will now fuse with this phagosome and squirt those granules in there. So because of that lysosome granules going with the phagosome, so now we have a phagolysosome now. So now the contents of the lysosome are emptied into that membrane-bound space now. So now we go to digestion, which is the 6th step. In other words, those lysosomal granules contain hydrolytic enzymes, and that means they will actually break down and digest the microorganism. Just totally dissolve it apart. So the last phase is when we spit it out. That's when we have excretion. So the phagolysosome goes to the outside of the cell and squirts out the trash. It's all digested, no good. Done, you know, it's dead. So the whole killing of a pathogen, we use this fancy chemical structure here, and it's a whole calculation reaction that occurs is by the hexomonophosphate shunt, hence the HMP, and that will convert NADP to NADPH. And what it's doing is converting oxygen. So oxygen from hydrogen peroxide to hydroxyl. And what it's doing is called an oxidative burst. It's actually destroying. It's like a, you've heard of antioxidants. Well, oxidants are there to help us destroy this bacteria, and this is how it does. So it's an oxygen-independent pathway. So the pH of the cell is changed now, which allows us to activate those lysosomal, those lytic agents, enzymes to actually destroy the bacteria itself. So it's not important to really know all the different steps, just know that it's a HMP that's converting NADP to NADPH, and oxygen is involved, hence the oxidative burst to destroy that particular pathogen that has gotten inside of the cell. Now, the other type of cellular cell that we talked about with lymphocytes was the natural killer cells, NK cells. So these are nonspecific. They are also our first line of defense against cells that have either tumors in them, some intracellular pathogen in them, or they're infected with a virus. In other words, our body is able to recognize this. So it recognizes these things that are damaged due to the things we just talked about because there's something different. It's altered. The cell looks different, and the natural killer cells, the NK cells, can see it. And what's good about this is that it's part of the innate system. It doesn't have to remember it. It just sees it, recognizes it, and it will then, the natural killer cell is activated. The NK cell will start squirting out certain chemicals, cytokines, and that will recruit T cells to come to the area, which then will cause more granulocytes and macrophages to come to the area and destroy it. So it's a multifocal approach to try to destroy it. So the natural killer cells, as you can see here, has a mechanism of cytotoxicity. So it's looking for the natural killer cell, it's got to have the target cell, and it's looking for this little thing on here, that little 3 green, 1 red thing. That will be an inhibition. In other words, it's saying, hey, this is a normal cell, do not touch it, so there will actually be no killing. And cells that are part of us have this little thing on, and we'll talk about what that little thing is a little later. So we don't kill our own cells, and because we don't want to do that. We want to just kill the things we really need to kill. Alright, so here is a natural killer cell. It's becoming activated because remember we said on this particular cell that's virally infected, it's got stuff on the outside, and our body recognizes it, so the natural killer cell kills it. Apoptosis, killing of the cell. Here is another cell, and it has an antibody. That's that Y-looking thing on it. So it says, oh, I need to kill this too. So it kills it too. So it's got activation from external things here or an antibody coded on it. So the innate lymphoid cells out there, they develop from the CLPs but do not express markers of the lymphoid lineage. So they're a little bit different looking, these particular type of cells. They're found mainly in the mucosal sites, and they're actually will release cytokines such as interferon and gamma in response to certain infectious agents out there. So they sort of regulate the immune system so we don't go overboard, or we do just enough out there. So in summary, the innate system is a normal defense mechanism that everybody has. It is not very specific. It just anything foreign kills it. We don't need prior exposure, and it will act the same way for every single foreign thing that gets in there. So we have external things. Remember we talked about our skin and all the different secretions that we have on there to protect us. And then there's internal defense mechanisms that we have out there. We talked about the phagocytosis. All the steps you went through that. How neutrophils, macrophages, dendritic cells do that. We have acute phase reactants like CRP that help us with phagocytosis. We have normal reflexes to try to get rid of it, pathogens. We cough and sneeze, hiccup, tears, salivate, all that kind of stuff. And for us to recognize it, we actually see the inflammatory process. And you've all had all this happen to yourself. So if you get down to the nitty-gritty, there's PRRs, that are molecules on our cells that will recognize the things that are on the pathogens, the bad stuff. And they're called PAMPs, pathogen-associated molecular patterns. So the PRRs recognize the PAMPs, because we want to make sure that when we try to kill something, we don't kill ourselves. We kill the non-self, the bad stuff, not our good stuff. Okay? So the acute phase reactants, they're just proteins that are soluble, floating around in our serum. And when we get an infectious process, they really increase quickly. And we actually do blood work, and we can analyze for these things, and that will tell us that. So that tells us that there's an infectious process or injury that could be occurring at the time. So our body, uses the inflammatory process to help us destroy, a pathogen that's coming in. And remember, with inflammation, we have the red swelling pain that occurs with it. That's how we can determine what's going on. I mean, that's what you look for. I mean, you can tell when you got something inflamed or infected on your arm or skin. So inflammation is, vasodilation, vasopermeability allows the macrophages, neutrophils, whatever, to migrate to the injured area and start doing phagocytosis and kill them, using the HMP. The hexose monophosphate shunt is a oxidative burst, so it's an oxygen-dependent and independent mechanism that is used to kill bacteria if we need to. So the natural killer cells are also part of the innate system, and they can actually kill targeted cells. In other words, there has to be a virus inside of that cell, or it has to have, like we showed a picture of an antibody attached to it, but it's able to kill things without prior exposure. So it's floating around in there. So it is actually the link between the innate and the adaptive immune system. So it's a link between the specific, which is adaptive, and the non-specific, which is the innate. Because the innate lymphoid cells, remember, they secrete chemicals, cytokines, chemokines, that tell other innate cells, hey, let's get going. We might need a more specific immune response, which that is actually called the adaptive system. So it is definitely a team approach in trying to destroy any kind of foreign thing that's out there. Alright, so that's the end of Chapter 2. So when you're looking at Chapter 2, you can make sure you read the chapter and look at the pictures. We went through all of those. There is a summary at the end and a study guide for you to look at. That's very important. There is some case studies, and of course there's review questions with the answers in the back of the book. Very good for you to look at so you can sort of review things again. We'll be doing a CRP lab. We're actually doing an in vitro phagocytosis lab, so you can actually see all this occur. But as you can see, it's an entire team approach. Your body has to work all together for it to protect you. And when you think about it, it's like, how do I get sick? I mean, there's multiple steps involved. There's multiple ways, we protect yourself, but it's easy for one of them to decrease. And we'll talk about how that occurs in later chapters. So take a look at this. If you have any questions, make sure you reach out to me. Emails, texts, in class, whatever you need to do. All right, well, thank you very much.